It's 10 o'clock, so yes. So just to remind everyone, this meeting will be recorded, so you can use it on our website. I would just like to begin by acknowledging the traditional custodians and owners of this land, the Wurundjeri people of the Kulin Nations. And these are the traditional owners of the land where I'm today, which is Parkville. I want to acknowledge the custodians of the lands you might be joining me from today and pay my respect to elders past, present, and emerging, and extend that respect to any Aboriginal elders who might be present today from other communities. Welcome and thank you for joining us, Professor Danev. This is our second of our CCE-MMP seminar series, and Rado has kindly agreed to join us today from Japan. Professor Danev graduated solid state physics at the University of Sofia in Bulgaria, and during his PhD, he worked on the development of phase plates for electron microscopy with Professor Nagayama in his lab in Okazaki, Japan. He published the first phase plate application in cryo-EM single particle analysis and cryotomography. And in 2011, he became a group leader at Max Planck Institute of Biochemistry. In the same year, he was awarded the Burton Medal of the Microscopy Society of America. And thereafter, he led academia industry collaboration, which resulted in the development of the Volta faceplate. For his research in 2017, he was given the Ernst Rösker Prize of the German Society of Electron Microscopy. And since 2018, he has been at the Graduate School of Medicine at the University of Tokyo. His current projects involve the cryo-EM studies of GPCRs, and he collaborates quite strongly with us here at Monash University with forays into cryotomography and more generally at method development in cryo-EM. So thank you very much for joining us today, Rado. A little bit of housekeeping before we begin. If everyone could keep themselves on mute and save questions to the end of the seminar, feel free to pop questions in the chat and I can read them out if you don't have a microphone. Just whenever you're ready, Rado, I'm going to turn my mic off and my video off. Thank you.
Thanks, Isabella, for the long introduction. I should have submitted a shorter bio, I guess. Share screen. Okay. And now the slides should be on and you should hear my voice as well. So thanks again for the invitation to the CCE-MMP. I hope I pronounced that correctly. It's a great thing to hear about this center, and also it has been great time together with Patrick working on GPCRs. And this study is actually part of our long history in cryo-EM investigations of the GPCRs. You see here some of the original or the pioneering members of this project. It seems like yesterday, but it's been already 5 years since we began the collaboration. So Patrick and Denise have led the project, and Mariam and Lynn, you see here on the right, they were the original experimental pioneers. So Lynn managed to express and optimize the GPCRs for the cryo-EM sample preparation, and then Mariam prepared the samples and did the observations. It was in 2016, I think, when we solved the first structure. Here on the bottom, you see Matthew, who is pushing the performance limits of data processing and getting these amazing results from even, data that is even so-so. And Cindy and Rachel and Sarah are working on the expression amplification. Now, I know that there are many more members in the team already at Monash, but I haven't met, unfortunately, or haven't had a chance to meet all of them. Hopefully someday we could visit each other and get to know each other better. So there is probably no need to introduce GPCRs to the audience, but a few years ago I knew nothing about GPCRs, and I guess this is as far as my knowledge goes at present. So they are the largest membrane protein family in the human genome with more than 800 members. Approximately half of them are olfactory receptors, so they're responsible for taste, vision, smell. And also neurotransmission and many other important physiological functions. So, and because of their broad range of signaling roles, they are also implicated in numerous chronic diseases. You can see here diabetes, heart disease, asthma, neurodegenerative disorders, and migraine, and many others. And because of their broad spectrum of physiological function or roles, they are also the largest group of targets of FDA-approved drugs. So they have a high pharmacological importance. Unfortunately, they are also notoriously difficult to crystallize, or in general for structural determination. Therefore, it took quite a while until we started getting really high resolution and nice structures. But in this talk, I'm actually gonna guide you through this journey. And I also touch upon some very practical details because there was hopefully a bit more time than usual. And I hope this will help people who are getting into cryo-EM at the moment. Here you see a plot of resolution versus molecular weight. It's probably dated already. I grabbed it from the Gatan website. But you can see in general that most of the cryo-EM structures that have been determined to date are quite heavier than GPCR. GPCRs at the ternary complex with the G protein is around 150 kilodalton. And you can see that most of the published or deposited structures in the MDB are heavier than that. So a few years ago, anything under 200 kilodalton was considered very challenging, and that included GPCRs at the time. And therefore, Patrick contacted us at the time we were still working on the faceplate developments to try and observe the GPCR they had at the moment, the calcitonin receptor, with the faceplate, so that we could probably have a chance to determine the structure. Now, I don't see the titles of my slide. I hope I don't mess up anything. But so the first thing we tried was the calcitonin receptor. And I think it was even on the first data set that Mariam collected, we got the structure. It was at 4.1 Ångström resolution at the time, which was quite impressive considering that nothing has been able to be done before we tried it with the faceplate. And with this resolution, the modeling was successful based on pre-existing models from X-ray crystallography, I guess, and docking the backbone and some of the larger side chains gave us a nice structure. Now, the faceplate itself is a device that is, as in light microscopy, changes the contrast properties of the microscope, where we can see phase contrast even when we are in focus. Now, conventional electron microscopy on the left doesn't have a faceplate. It uses just an objective lens aperture. And to create phase contrast, we have to defocus the objective lens. However, this has a kind of a high pass filter effect. So to get the low frequencies and the strong contrast in the image, we have to defocus a lot, which then compromises the recovery of the high frequency and high resolution components. If we use a faceplate, like shown on the right, it enables in-focus phase contrast, meaning that we can collect data very close to focus and still get very nicely contrasted images as you saw in the previous slides, and then be able to reconstruct them to high resolution as we showed in some of our tests. Here you see a kind of a summary of the GPCR structure determination by cryo-EM, and this was by the end of 2018, I think, when I made this slide. Since then, I'm sure there have been many, many more structures. But at the time, we were still using the faceplate, and all the structures shown here, which have the title in red, were collected with the faceplate, and the ones in blue were collected without the faceplate. And the general impression from the data that was coming out and from our use of the faceplate in practice was that it really helps, because you can see the resolutions we were getting were slightly better, and also achieving those structures was not as challenging, it seemed, and then without the faceplate. Nevertheless, because so with the faceplate, we also had a few other kind of technical achievements. So the first cryo-EM high resolution structure of the nucleosome in collaboration with Sarah Sandin's group in Singapore. And a technical achievement, Maria managed to get a 3.2 Ångström structure of hemoglobin. It has a historical value in a sense that it was the second structure that was solved by X-ray crystallography in the 1960s. And it was very challenging for cryo-EM. So it is only 60 kilodalton, and it was believed that it would be very difficult to determine. But with the faceplate, we managed to determine the structure in 2017. Later on in 2019, there was another paper of streptavidin, a 52 kilodalton complex, and it was also considered, it is still challenging, but at the time it was considered even more challenging. Nevertheless, other groups were pushing also the performance without the faceplate, namely Game Bladner and his team. They had the slogan, no faceplate, no problem. And they actually determined the structure of hemoglobin in 2019 at 2.8, if I remember correctly. I don't see the title and I don't know how to actually hide the Zoom. Hide video panel. No, okay. Let me, bear with me, please. Okay, I managed to get it. Good. So, yeah, so it was starting to be, I started to become conscious of, you know, are we really doing the best with the faceplate? And also, we never have done actually one-to-one comparison to show for sure that it helps and by how much it helps. So after moving to the University of Tokyo and getting some new microscopes, I decided that this would be a good chance to actually do this. And we collected in 2018, one of the first data sets actually we collected since renewing the collaboration after my move to Tokyo was half and half. So basically half of the data set was collected with the water faceplate. You see on the left here, much higher contrast, and another half without the faceplate. Now, processing the data, unfortunately, showed that the faceplate doesn't really help. And you see here some features of the side chains of two of the helices. So the blue one is without the faceplate, the red one is with the faceplate. The difference in resolution was 0.3 angstrom, so 2.7 versus 3.0, which is around 10%. And visually, you know, looking at the features of the map, they are not that different. But nevertheless, you know, the faceplate in practice is more difficult to use, especially for people who have no experience with it, and makes the whole experiment, it increases the risk of something going wrong with the experiment. We also quantified the overall performance using the so-called B-factor plot, and also called the Rosenthal-Henderson plot. Basically, you plot the resolution in a 1 over resolution square. So it's not a linear axis, but you know, reciprocal squared axis versus the logarithm of the number of particles. And then the theory says that it should be a linear dependence. And the slope of this curve shows what we call in electron microscopy a B-factor. Now, this B-factor is a bit different than the B-factor in X-ray crystallography. In cryo-EM, the B-factor represents the overall envelope function of your data in reciprocal space. So basically, it incorporates all factors that may impair the performance of the experiment into one global quantity. And the higher this number, the worse the performance. And you see here for the water faceplate, the slope was lower or less. And therefore the B-factor was higher, and that means that the performance is worse. So basically you can see from the plot, the lower B-factor means that the curve kind of increases slower with the number of particles, and therefore you have to increase the number basically exponentially to get to the next resolution level. Therefore, the number of particles required to reach a particular resolution at some point kind of rises exponentially, and one shouldn't try to improve their experimental performance in general by collecting more and more and more particles, because there is a very strong effect of diminishing returns at some point. So this experiment went pretty well in a sense that we had both a structural result. So this paper produced a nice structure. I believe this was it was not calcitonin. I forgot. Anyways, so there was a very nice structural result, but also a technical kind of study result, in a sense, comparing two different experimental conditions. And from that, I decided, let's continue doing this for the other factors. We are kind of wondering about how much their contributions are. So zero loss energy filtering. In the past, some companies were telling customers that no, filtering is not important for single particles because you're in thin ice, it only matters for thicker ice. Of course, they had an agenda. They didn't have a hardware at that time. They didn't have their own filter. Nowadays, this company has their own filter, and of course they trumpet the Trump and the song that it helps a lot. Anyway, so we decided to test this and see if it really helps or not. Also, the objective lens aperture, which is also being a subject of debate in the community. Do we really need it in single particles? Does it help, and by how much? and so on. So included that in the comparisons. The focus magnitude. So we tended to, and we still tend to use, at least in my group, a bit lower defocus values than other places, believing that it doesn't really hurt the data, even for relatively small particles like GPCRs. The total exposure, meaning how many total electrons you expose the sample to. At some point, of course, the high resolution components are gone after radiation damage progresses. And then you basically collect, the last information in terms of very low frequency components, basically just to increase the overall contrast of the particles. And finally, the specimen grid type, that was a kind of a side result from these studies, because we kind of initially inadvertently, but later on, completely switched to the gold type grids, and they provide quite a big improvement in the data. So here is the grand summary of all these individual investigation of all these individual factors. So the Volta faceplate, it reduced the resolution by 0.4 angstrom. Now here, what you see plotted is the resolution from 100,000 particles. This is to make things a bit more comparable, although depending on the sample, depending on the many factors in practice, things are not comparable between data sets, but within each data set, because it's from the same grid and the same sample, basically this is as close as you can get to a fair comparison. In any case, lower resolution with the Volta faceplate and also worse B factor. So unfortunately, it didn't help. And since we got this result, we stopped using it in single particles. Zero loss energy filtering provided quite a substantial improvement in the performance. So 0.26 angstrom. So negative value means kind of lower resolution number, so higher resolution actually. Objective lens aperture had a almost unnoticeable effect, although it blocked these crystal reflections, which you see sometimes in the images from the polycrystalline gold film. Defocus magnitude, so here more than 1 micron defocus provide a slightly better performance. So in general, yes, it seems that the higher defocus helps, particularly during classification steps, where the low frequency components are helpful to classify, I guess, damaged particles or kind of disassembled complexes. And higher exposure had also a noticeably positive effect. So instead of 40 electrons, this is comparing 40 versus 65 electrons per square angstrom, and it seemed it helped. Now in practice, we can in principle expose indefinitely, because we are collecting movies and the dose weighting, or you can just cut off the frames at some point. But from a practical point of view, this requires a longer exposure time. So basically you sacrifice a bit of throughput, but the difference is not that big, because from 40 to 65, maybe instead of 2, you have to do 3 seconds, but there is also many other overheads during data collection. So in any case, it's a compromise which one has to make, I guess, for themselves. This is also in this plot, the grand summary of the results we got from many complexes. I'm sorry, I'm in quarantine and I have to report to this application when they call me to make sure that I'm still at home. So I will say I'm here. Okay. So in this plot, you see the single particle results from between the end of 2018, when we started at the University of Tokyo, until April of last year. So basically a year and a half. Since then, we have, of course, more structures, but this is, the data when we prepare the manuscript. And you see here, there is a general trend of the resolution improving. So we started at around 3 angstrom, and I was kind of determined in 2019 to reach 2.5. And it so happened that we actually reached 2.1. So it was over our expectation at the time. And then you see here the big change happened when we switched from carbon film support grids to gold foil. And this happened kind of by chance, because we've had apoferritin results at 1.6 angstrom using the carbon grids. And I didn't expect that those, the support itself will limit GPCRs that were at 2 or 2.1 or 2 above resolutions. So basically, I was a bit, if we can reach 1.6, then 2.1 is, completely safe. But switching to gold grids actually improved not only the beam-induced motion, it also improved the quality of the grids themselves. So because the grids and the quality in which they are manufactured seems to be much better, they don't have this lip on the side of the hull. And of course, the reduced motion and the film itself is more robust. So during plunging, even if you blot a bit longer, it doesn't break on the squares. You see here on the left carbon grid that we were using before switching to the golds. And we switched by accident because I ran out of the carbon film quantifoil. So I had a box of the ultra aerofoils, and then I tried them. And after seeing the first few data sets, we were all sold on the gold foil grids. So you see here the carbon grids, more broken squares. Of course, this is a larger mesh, so bigger square, so less protected by the grid bars. On the right, you see the gold foil grid. Now, it's very difficult to judge the ice thickness, or more difficult to judge the ice thickness on the gold grid because of the high contrast of the support film. But trust me, the film is the ice thickness distribution is much more uniform, and we get many more squares with a thin kind of uniform thickness size, which is very important when collecting the data. The ice thickness and the particle distribution are the most important factors. These are histogram plots of the motion, beam-induced motion. In this case, I plotted the displacement because the values that come from RELIAN that represent the summed motion, basically frame to frame, the absolute values added together, seem to be biased or influenced by noise. So if you have a lot of frames in your movies, or kind of plenty of frames in your movies, then the noise kind of between frames starts to accumulate, and then you get less, the plots I made were less, showed less of a difference, let's say. But plotting the displacement, which is actually from the start of the movie until the last frame, just the difference in the positions, showed a much clearer picture. So you see in the early displacement, the gold grid, the blue one, shows much smaller motions, and overall the total displacement was also lower. So definitely less motion with the gold film grids. And looking back at the plot of the reconstructions, we see, the general tendency of resolution improving. So there was a hope and kind of, enthusiasm to, can we push things further? Can we go under the next psychological barrier, the 2.0 angstrom? And last year, indeed, we got a new microscope, the Titan G4, and it was installed in a place where it is there is much less external disturbances, external fields. The first machine was installed in a building where there are elevators like 15 meters away. So we had the magnetic field canceling system, but nevertheless, judging from the movement of the energy filter slit position, it seems that we have quite a not as favorable environment. But in the new place, it was a separate small building dedicated to microscopy. The environment was much quieter. Also, the machine is much better. The stage seems much more stable than the earlier stages. The box is more tight, both in terms of temperature and noise. So it isolates from the environment. That's also very important. And the machine as a whole had a much smoother and kind of stable, gave me a much smoother and stable impression. So on one of the first data sets already we collected, we got sub 2 angstrom resolution. You see here the CCK1 receptor we published earlier this year at 1.95. And we have one more structure which is not yet published, but you can see the features here, both in the receptor and in the G protein. We see the holes in the aromatic residues, and also hints of the pyrrole, the pentameric ring in tryptophan starting to show up. So indeed, we are sub 2 angstrom. And of course, this is for the ternary complex. This is for the highly optimized, nicely distributed sample at high concentration. These kind of performance, we hope will kind of expand to the more challenging targets that Patrick's group is now moving on to. I will show about this a bit later. Let me just give you a few details about the data set. So with these ternary complex, sub 2 angstrom structures, of course, ultra aerofoil grids, kind of high concentration to get a dense packing of the particles in the ice. typically we have more than 50% coverage of the image, I would say, by particles, but not too much. So if you have, like here at the top part, if you have too many particles, they may start to actually mechanically obstruct each other or maybe push each other and deform the micelle or something like this. So I would say between 50 and 75% by area coverage of the image seems to be optimal. This data set was collected without the objective-less aperture because at the time I didn't see or didn't believe that it will make much of a difference, especially if you want to reach high resolution. Sometimes the aperture can actually introduce higher order aberrations because of charging, and therefore one has to be careful. So grid score. So I have a scoring system where during screening, I will explain later what the score is, but it's important, I think, at any facility to have some kind of traceability or ability to quantify the effect of different things, to develop some kind of scoring system for the samples. Therefore, you can trace and check what had an effect and in what sense. For the microscopy, I went one magnification up from the one we typically use. So instead of 0.8 angstrom per pixel, we are at 0.65. This should improve the DQ performance of the camera. We use the beam image shift acquisition in cryo-EM with a K3 detector in CDS mode. We still have the issue though that the first frame of the movie has a lot of systematic noise, a lot of pattern noise. So we have to throw away the first frame. So I hope that one day this will be fixed with the firmware update. The last attempt to fix it on our microscope didn't succeed. Actually, it made things worse. So they reverted the firmware, but we still get more crashes during data acquisition. I hope this will be fixed someday. We also used a bit narrower energy filter slit, 15 EV instead of the 25 EV that we were using on the Cryos 1. This is because the new microscope was, as I mentioned, in a very quiet environment, and we could go with a narrower slit without having to do realignment of it overnight. Now aperture, defocus range, you see here 0.6, 1.3. Typical defocus range for me is 0.5 to 1.5 basically, but this is the actual range that was measured after fitting the CTFs. And other than this, it was one image in the center of each hole. And you see here 92 frames, quite a few frames, and 70 electrons per square angstrom. So typically I'm aiming at 0.8 electrons per square angstrom per frame. That's kind of a rough target. After particle picking, there were around 4 million particles, and after classification steps, around 600,000 left. So this is the typical kind of filtration, reduction during filtration of what we get with the datasets nowadays. Now, another very challenging target that we are able to determine is apostate receptors. So basically receptors without the G protein associated, both without ligand and with ligand. This was for the CGRP receptor, and you see the resolution is not as high, of course, as the ternary complexes that are bigger and much easier to align, but it is still quite an achievement because that opens the doors and shows that cryo-EM can also do the receptor-only structures, the inactive state structures, which have been, prerogative of X-ray crystallography until now. They still are more successful, I guess, in crystallography with them because I guess it's much easier to crystallize this receptor only than the ternary complex. Showing a plot of, again, resolution versus molecular weight. This was a more recent plot published in a review last year by Mengyu Wu and Gabriel Lander. So you see here that there are not many structures in the EMDB below 100 kilodalton. So here is 100 kilodalton, 150. This is where the ternary complex is. And receptor only is at around 74, 70, depending on the weight of the ligand, I guess could be a few kilodalton less, more or less. So not many structures in this range and very few actually membrane proteins. I don't know if there is another non-symmetric membrane protein. There is one C2 symmetry ion channel, I think, from the SARS-CoV-2 virus in this range. Even more recent review by Gabe Lander and Robert Glazer, where they kind of analyzed or discussed the cryo-EM of small particles. You see here actually that last year there were around 20 structures deposited, and this year around 50 already. So there is definitely a growth in the number of structures below 100 kilodalton done by cryo-EM, but they are still quite challenging depending on the type of protein. Most of those, I think, are RNA complexes, ribose switches, or soluble proteins basically. And if there is a nucleic acid there, it has also higher density and is also more radiation hardened. So it probably helps to do these smaller complexes if there is RNA or DNA. These are the dataset details for the receptor-only structure. So again, quite a high concentration to get this nicely packed in thin ice distribution. So basically the idea is to have a single layer of molecules, kind of keeping the ice from collapsing. And this grid has a very high score. So both numbers are independent. So it's not 97, it's 9 and 7. So the first one is the grid quality and ice quality. The second one is the particle distribution. For these data sets, I didn't go to higher magnification because I was not, I didn't believe they will go to 2 angstrom or close to 2 angstrom. And, you know, that was the case. So they were at around 3.2. Later, somebody was reprocessing the data sets during a challenge that was organized by Patrick and Matthew. And I think they got 2 point something, 2.9 or 2.7. Anyways, CDS was on again. This data was collected on the, I believe, the first cryos. So this data was collected before we got the new cryos. And you see the energy slit was wider. Nevertheless, it still worked out in the end. And also with the 100 micron aperture. So yeah, it probably also helped a bit. So nowadays I'm going back to using the aperture because with the gold foil grids, especially, it removes these bright spots you saw on the previous image. Again, lots of particles, and yeah, maybe 10% or less were left after the classification steps in the end. So there are probably a lot of damaged or you know, not well imaged particles. So lots of data is being thrown out, but it's important to end up with a nice juicy data set in the end, which has a lot of signal. Now I prepared a couple of slides which explain in detail the practical factors which I believe are important. So during sample preparation, I start the vitrobot at least 1 hour before the plunging, and I put the filter papers at that time because the filter papers have to be in this high humidity environment. We keep it at 4 degrees C, 100%, to get saturated in a sense, humidified. Because if they are dry, if you put them just before the plunging, during the blotting, because they suck out the water, but they are also very near to the surface of the grid, they can, my subjective opinion is they cannot act as a dehumidifier, basically sucking out the, making the atmosphere around the grid less humid, and that could, you know, increase the evaporation rate and make also things quite random with the ice thicknesses you're getting. Also use a scrub grid and put it on the tweezer and do a run of the vitrobot, a dry run, so to say, and carefully observe with the long blotting time, say 10 seconds or so, carefully observe if the papers are touching the grid uniformly on both sides in synchron, so not one touching before the other. If they're not properly centered, there is a way to center them. There is a special tool, ask your service engineer. And also if the grid is well kind of towards the bottom of the filter papers and well hugged by the papers, not you know, kind of hanging, and you can, you know, clearly see that only the bottom edge is touching the filter papers. That also introduces a lot of inconsistencies in sample preparation. We pre-wash the grids with acetone. This is just from the quantified times when there was still some plastic left on some of the grids. Glow discharge conditions, basically, I increase the glow discharge time until I see less and less pooling of the sample in the middle of square. So I will go back to the grid maps. So you see, if you in the grid maps, you see a lot of these kind of dark blobs in the middle of squares. That means there is a drop remaining, liquid drop remaining in the center of the square. That means there was no hydrophilic path for this liquid to drain through to the filter paper. So if you imagine the grid, there is a carbon on top or a gold on top. On the bottom of the square, the filter paper cannot get very close to the film. And if there is no path for the liquid to drain, then it just pools there. And it's not touching the grid bars because the grid bars are hydrophobic. Increasing the glow discharge time can make them a bit more hydrophilic, and also depends on the grid material. But in my experience, yes, by increasing the glow discharge time, this effect is reduced quite a bit. Don't go too high though, because with the gold grids, if you go too strong with the glow discharge and also with the carbons, you start to damage the film. And this is visible by the edges of the holes become rugged, so kind of jagged. And this means the gold film is starting to be etched or recrystallized during the glow discharge. I always use the same sample volume, the same blotting time, and the same blotting force. This is very much dependent on the machine, the with robot machine you have, or if you have a different plunger. So the blotting force is the thing you have to find what works for you. But once I had these parameters set, I don't touch them. So the only thing I touched with the highly optimized samples that Patrick's people are sending is the concentration. So basically, depending on the starting concentration, plunge 1 or 2 grids at the initial concentration, we get 10 microliter aliquots from them in PCR tubes. And it's important not to have too much because you don't want to waste samples. So just throw one tube, prepare samples, screen them. If they're not good, throw another tube. I thaw them shortly before plunging and then plunge the original concentration. If it's not too high, say if it's at 3 milligrams per liter, then maybe 2 grids. Dilute twice, so add buffer for 2 times dilution, plunge 1 or 2 grids, and then if it's really high concentration, say 10 milligrams per milliliter or even 12, I've gotten some samples, then maybe plunge a 3rd grid with 4 times dilution basically at this time. Then the screening process, we do the screening on the towel, so typically between 8 and 10 grids, depending on how many samples, and this takes 1 day. The procedure is load a grid, acquire a grid map. You see here the grid map. Then select squares which look to have good eyes in them. And with the cryo-EM script, we collect in each square 1 image in the center. And this is what the images look like. So you get both an image of the hole and the image of the particles at high magnification. So you see here, this square, for example, the eyes looks okay if you look at the hole, but the particle concentration was actually quite high. You see that there are, highly packed areas and also areas where there are obviously 2 layers of particles. In another square, the eyes looks very similar to this one, but if you look, we have a nice particle distribution with just a little bit of gap between the particles. So this would be ideal place to collect. Then based on these screening images and all these, for each square particle image, when we put the grid in the cryos, we also have this grid map open on the notebook computer next to the microscope, and I start collecting in the squares that show the best particle distribution. So I select 3 or 4 squares based on this. I select many squares, but I start selecting from the squares that have the best particle distribution. And typically, maybe 30, not 30, nowadays maybe 50 or more squares from 1 grid, but starting always with the best looking squares. Then we go to the high mag, start the acquisition immediately as soon as possible after checking the beam and a few other things to set up the acquisition, and start from the squares that had the best looking screening images. And once the acquisition is started, the nice thing with cryo-EM is that the acquisition is already running, but you can continue adding more squares kind of at your own pace based on these square maps that we collected in the cryos later. So yeah, basically there is no rush. Once you start the acquisition, there is no rush to select, carefully the other squares. I hope the TPU will have this functionality sooner or later. Nowadays, of course, they have the automatic addition of new areas. So you basically select 1 square or 2 squares that look good to you. And as far as I heard, it's able to, based on the, the ice thickness or the intensity, it's able to automatically add similarly looking squares to the acquisition queue. Now, moving on to more challenging targets, I think that's also Patrick's priority nowadays, not only the receptor-only structures, but also, for example, a structure without the proprietary nanobody 35. So this nanobody is, although it's not shown here in the map, it's located here between the alpha and beta subunits of the G protein, and it stabilizes the complex overall. I think it's not only making the G protein more rigid, but the whole structure becomes more rigid. And here you see two structures with and without the nanobody. Of course, with the nanobody, the structure was at a bit higher resolution. But with this study, Patrick's group showed that it's possible to determine structures even without using this proprietary nanobody. And of course, moving on to dynamics, nowadays with many possible tools, we first tried the reliant multi-body refinement. Of course, that's a bit more involved, it takes time. But nowadays with CryoSpark and the multivariability analysis, 3D variability analysis, it's very easy to run this kind of task. And in the case of semaglutide and taspaglutide that was published earlier this year, the consensus structure were almost identical. Practically, the models were very, very close. I don't know the RMSDs, but indeed, one couldn't see much difference between the consensus models, although the receptors have a different kind of signaling profile. And looking at the dynamics, it showed a more, I think, larger movements of helices 6 and 7, if I remember correctly, in the semaglutide. I hope I didn't mess this up. So I think it was the semaglutide that was moving more than the taspaglutide. And that shows that dynamics are actually as expected, play a very important role in the signaling profile and bias of the receptors. And another analysis that was done in the paper of the receptor-only structures. So there was also very nice data of hydrogen deuterium exchange mass spec, and this data shown as a color code of the model here correlated very nicely with again 3D variability analysis, showing that dynamics are corroborated by both of these techniques. And I think such integrative approaches will continue to grow in terms of also numbers of approaches people use to study their complex. And then of course, the more sources you have for information, the better, especially when both sources agree on a particular finding. So my vision for the future, or the things I look forward to in the future in pushing the cryo-EM performance even further, is the cold field emission gun. This is not that important at the moment, especially at the 300 kV acceleration voltage, because we are not at a 1.2 angstrom resolution with the GPCRs, but at 200 kV, already at, I think, 1.8 or 1.7 angstrom resolution, you hit the temporal coherence envelope with the Schottky field emission gun. So cold FEC, I'm really looking forward to. Better detectors, that's always the technology that has been, the major driver of the advancements in cryo-EM. So it started the revolution, and the detectors nowadays are also the most important part, so to say, in terms of throughput and overall performance. Laser faceplate, so because the water faceplate is obviously impairing the information transfer at high resolutions, the laser faceplate is the next step. Actually, it's the ultimate solution because there is no material in the beam path and therefore no loss of information at the high frequencies. Of course, there are many technological challenges there. I know I have run out of time. This is my last slide. So yeah, sample prep, very important, especially for the real biology, real pharmacology as well. So all these structures that I showed are, I think, were achieved primarily because of the efforts in Patrick's group and early on by Lynn and her followers on optimizing the samples. Screen your samples carefully, do negative stain. That's ancient technique, but it's very helpful. Automation is very important nowadays. And of course, advances in data processing, which also drove the cryo-EM revolution. So I'm grateful to Masaki Kawa at the University of Tokyo, who invited me to join the project of establishing a new facility there, and it has been very successful. Of course, the dean at the time, Kohei Miyazono. Yanagisawa-san expressed and generated the first this apoferritin that was so instrumental, and Nakamura-san, the facility manager, and Fabian, my new postdoc, who is working on tomography challenges nowadays. And thanks a lot, and sorry for being over time by 2 minutes.
Thanks for a great talk, Rado. You're actually not over time at all. We've got a bit of extra time scheduled in for questions.
Okay, I started hearing noises and I got, oh, there.
No,
okay.
I can't mute other people, unfortunately, but if everyone could keep themselves on mute,
anyways,
unless they're asking a question. But thank you so much. It was a great talk. Are you happy to take questions? Are you happy to take questions, Rado?
Sure.
Okay, so if anyone has any questions, please pop your hand up and I can call on you. Or else we've got a question in the chat from Josh. Do you want to read it out, Josh, or would you like me to?
Yeah, I can read it out. Thanks, Rado. That was really great. Thanks for sharing your experimental process as well. I got a lot out of that. I was wondering about the faceplate and I guess why you think that there was a slightly worse resolution when you did reconstructions with the faceplate as opposed to without. And, you know, I've been reading about your work on this, trying to figure out what the kind of theoretical explanation would be. Is it due to inelastic scattering or loss of signal as it passes through the faceplate, as you've described before? And I guess as a follow-up to that, do you still see a role for the faceplate and single particle cryo-EM?
So with the faceplate, originally, when we were investigating the water faceplate, we saw that there is a 20% loss of information, basically, because the fume of the faceplate scatters some of the electrons inelastically, or probably some of them also elastically. They kind of don't contribute anymore, don't carry anymore the information. And the thinking was that you lose 20% over the uniformly across the frequency range. But later on, it, after more investigation, it turned out that maybe the higher frequencies are affected more. We don't know why. Now, this is a mystery. We published a paper with Bob Glazer and Bart Bause recently called the DQE of the water faceplate, and there is no answer why this happens. There are experiments showing that indeed the higher frequencies are attenuated more. I still see a role of the water faceplate in cryotomography, so we are currently working on some things that are quite challenging, and also we are trying to look at morphology there. And for morphology, higher contrast is indispensable. I mean, it helps a lot. Nowadays, you have these denoising methods where you can make things look like faceplate, but still they don't give you a signal. They don't give you more signal. They just make things look better. So denoising the faceplate images actually looks even better. So we are coming back to the faceplate in tomography. And I see the future for the laser faceplate. Mm-hmm.
Thanks.
Did that answer?
Yeah.
So we have a quick question from Hongyi, and then I might go to Isabel. So Hongyi Shu says it's midnight where he's coming from. So he would like to know about how much benefits will CS or even CC correctors bring to cryo-EM. If you had any thoughts on that.
The CC corrector, I don't see at this stage. I know Richard Henderson and Chris Russo are quite enthusiastic about it. The point with the CC corrector is you don't do zero loss filtering anymore. You just kind of rescue the elastic electrons, but this eventually may give you a little bit more signal at the high resolution frequencies. That's what they showed, but you lose the low frequency contrast basically by not zero loss filtering. So we have to see the first experiments, I can say, but I'm not too optimistic about this in tomography especially. So single particles, maybe, yes.
Cool. Isabel, did you want to ask a question?
Sure. Very nice work, Rado. Really, really amazing amount of structure you are showing us here. My question was regarding when you evaluate the resolution and indicate the performance factors that improve your resolution. And if you use for your shell correlation, you are really comparing the consistency of the data. And if you see an improvement of 0.2 angstrom, is it really, really meaningful? Or is it just because the data is a bit more consistent because, for example, you have more dose? I wonder if you could comment on that.
Yeah, that's a really good point. So the differences are really small, of course. And that means that I would say that's a good sign, meaning that the factors that maybe had a really small experimental impact, like the aperture, didn't show erroneous signal in a sense. The differences were calculated based on B-factor plots. So first make the B-factor plots done with random particle subsets. So it's not just taking the final resolution of the subsets we got. We also did these random subsets. So it's a bit more, I would say, quantitative. And yeah, the differences may be insignificant in terms of modeling. Say if you want 0.2 angstrom resolution difference in your map, that probably won't affect much the quality of the map or the ease of modeling, but they accumulate. So if you combine several factors, you may get 0.5 angstrom difference. Then that's significant in practice. And of course, the gold grids were the biggest factor in a sense that they, gave us this 0.5 angstrom improvement overall and more consistent sample preparation. So for samples that are challenging, so if you have something that you have to collect in thicker ice or something that you have to collect with tilt, really challenging samples, then these small factors probably won't affect the final outcome that much. These factors get important when you already have optimized the sample and you're lucky to have particles which are randomly oriented in the ice. So they won't rescue the sample. Of course, energy filtering, zero loss energy filtering is always a must, I would say, especially with thicker ice or tilted samples where you also kind of increase the thickness of the sample. So yeah, hope that answers your question.
Amazing. Thanks, Isabel. I think Jack was next and then Danev. Jack?
Hi, Rado. Thanks for the talk. It was very informative. I just had a quick question about the Volta faceplate. So I understand it's a physical screen sitting in the microscope. Does that have to be kept in, I suppose, a locked alignment with the sample grid itself, or does rotation of that sample grid when you're trying to look for different angles and things like tomography, does that introduce sort of a skew in terms of electron interference?
So I guess you're asking if moving the sample or tilting the sample affects the performance of the faceplate. Is that what you're asking?
Yes, and if you see sort of a difference between, I suppose, parts of the grid closer to the center of that tilt axis rather than further out.
So one effect which is crucial and I think is quite detrimental in tomography, especially, is sample charging. So not the Volta faceplate, but the sample is electrostatically charging, and then it starts to act like an electrostatic lens. And if it's not tilted, then the beam just kind of converges faster and then basically defocus on the faceplate. That hurts the contrast. But if you tilt the sample, things become even worse because the beam then can be deflected to one side and basically moved on the faceplate and you lose the phase contrast. So sample charging is very damaging to faceplates in general, not just the Volta faceplate, any faceplate that has, phase shifting something in the middle of the diffraction plane will basically be affected by that. Apart from this, during tilt series, if the sample is tilted and if you're very close to focus, some parts of the sample may get over focused. So that's not good. So there should be always some defocus applied so that no part of the sample gets over focused. In principle, you can use the over focused areas, but dealing with them in practice may be a bit difficult. I haven't done tomography in these conditions, so I don't have experience, but I've heard from people that you shouldn't go over focus.
All right, thank you.
Mm-hmm.
Cool, thanks, Jack. Danev?
Hi, Rado. Thanks very much. It was really nice.
Right.
I very much enjoyed. You kind of answered my first question. I had two questions, so the first question was kind of answered in the last question. The second one is, now with the sort of development of fast incremental tilt series data collection, is there a sort of way to include faceplate? Will that be possible, or it's just not compatible with fast tilt series data collection?
On the contrary, it's actually more compatible. So during the fast tilt series, the idea is you don't do focusing and tracking. You just collect the images, and from the images themselves, you predict the motion of the sample and maybe the defocus. And that's very nice because one of the things that were hurting the Volta faceplate during acquisition is when you do this tracking image or focusing image, when you go off axis, that can, if the alignments are not done perfectly, that will hit the faceplate on a different position and may distort the phase shifting area. So by just collecting images only, and let's assume the sample is not charging, actually the faceplate will work much better.
All right. Okay.
I don't think we have tried it yet, but yeah, definitely, I think it would be better for the faceplate to just collect exposures and no tracking and focusing.
Yeah, I have not heard anyone trying this scheme with the fast incremental data collection scheme. Thanks for your comment.
We'll try it.
Thank you.
I think Eric had his hand up for a while and I missed him. So if Eric wants to ask his question.
Hi, Rado, very nice talk. I guess, have you compared the different detectors from these manufacturers and maybe another?
Yeah, so we had a paper recently on actually when we got the new Cryos last year, one of the first things I did is collected apoferritin data sets, of course, to make sure that everything works. So I don't know if you have seen it. So we have K3 with the filter and K3 without the filter. I mean, it's still behind the filter, but I retracted the slit. You know, that was the fairest. And you have a Falcon 4 before the filter. So I collected, you know, apoferritin data sets from the same grid. Basically, they have identical performance per pixel. So I didn't see in my test, even, you know, some people were saying, but if you retract the slit, the filter can introduce some distortions or something. I didn't see any detrimental effect. I don't know why putting the slit out will change the aberration performance of the filter. It didn't change, obviously, but for me, I got identical results.
I was more talking about small protein like GPCR more than apoferritin.
Okay, sorry. No, I haven't compared that with real protein. We are just, yeah, we have the workflow already with cryo-EM, and the Falcon 4 when it came out last year, and it's also not behind the filter on our microscope. We had to use CPU, and you know, with CPU the throughput at the time at least was lower for us, and yeah, also no filter means I would guess, yeah, one has to do a data set without the slit for the K3 and compare it with the Falcon data, which won't be optimal for the structural study that is going to be using this. So I haven't done it now.
I guess we can try it both ways.
Or you guys can try.
Yeah, thanks.
Yeah. We might take a few questions from the chat. We are coming up to 11 o'clock though. So are you able to stay past 11, Rado, or do you have to run?
Sure.
Okay, cool. So Yufeng Zhao has a question. He says, I missed the step about pre-wash the grids with acetone before blow discharge. What is the purpose of this step?
So the pre-washing was in the past with the quantifoils, the way they were made, or I guess still made, they make a plastic film with some kind of template. Then they evaporate carbon on top of the plastic, and then they dissolve the plastic. But in some batches, the plastic, the amount of plastic that remains varies. And so I made it a kind of a standard step to just put the grids on a filter paper with the film up in a Petri dish, glass Petri dish, and then pipette in a milliliter or so of acetone to very well soak the paper, so that when the paper is soaked, actually the grids also get kind of soaked into the acetone. This dissolves any remaining plastic from below the grid, and then you just let it dry to evaporate the acetone. So it basically recedes down into the filter paper, and it's I guess just become a traditional step of the workflow. I can probably omit it nowadays, but I keep it there because this is what works.
Sounds good. William also had a question in the chat before I get to you, James. He said, can you comment on CDS versus non-CDS on the K3? I see you are using CDS in your beta structures.
Mm-hmm. Yes, we use it as when we started using it, we get the subjective impression it was a little bit better. I planned to test CDS, non-CDS one-to-one, like I did with the other factors. Unfortunately, because we don't have this issue of the first frame pattern noise fixed yet, one would say, it's not working as it should. So I never got to testing it. But from other people's comments also, there is subjective impression that it provides benefit. Although there was a paper, there is a paper from UCSF, I think Dave Egert and the Yifan Chen's group, they published a paper showing that there is definitely an improvement in performance with CDS. Of course, it requires you cutting down the exposure rate in half. So, you have to use maybe twice as long exposure time. So there is again a compromise. But in our workflow, it seems to work well. So yeah, I can't say for sure though. don't put, don't bet on this.
All right, cool. I think James has a question.
Hi, Rado. How are you?
Hey, good.
Good to see you again. Hey, really great talk, and I always like the analytics. So I have kind of a couple questions, wondering if you'd looked at two things, sort of the aberration-free image correction, of course, again, related to speed versus quality of the data, and then also multiple shots on a single hole. Wonder if you have any analytics kind of that, or have some feelings about kind of those sort of two techniques there.
So the aberration-free image shift is EPU feature basically, and it's I guess it's great because it improves the throughput by a lot. And because I don't use EPU, I'm using the cryo-EM version, which is called beam tilt compensation. Basically, when cryo-EM does image shift, it compensates for the beam tilt, which is exactly what EPU does. It's just a different. The company had to put a different acronym, right? They couldn't just. And yeah, it works great. Of course, we are using it. Although not with as many holes in cryo-EM because there is no intelligent selection of sub-areas. So if you expand the pattern, if you make the pattern larger, so instead of 3 by 3, if you do 5 by 5, then yes, you'll get higher throughput, but then your utilization of the area of each square will be less because you have to have a fixed number of these patterns in a sense. Hopefully that will improve with newer versions or better, I guess, functions in cryo-EM. The other question was about
multiple shots.
Yeah, multiple images. Yeah.
Kind of like
so. Yeah, we are collecting nowadays recently with 2 images per hole just because of the magnification and the shape of the K2. You know, it's a bit rectangular, so you cannot fit 4 easily unless you go to higher mark. There you can do 4 in principle, but with the gold grids, I would say. In terms of beam-induced motion, there is no detrimental effect. There is a little bit. I still see, you know, perpendicular to the edge of the hole. If you are close to the edge, you should always touch a bit of the film with the beam. That's important to discharge the eyes. But it doesn't hurt, I think. So yeah, multiple shots per hole are definitely okay. What's actually the decision-making factor is looking at the sample. Say the concentration is pretty low, then you don't have, for example, particles in the center of holes. Then it definitely should collect, you know, a few images around the edge. And if, on the other hand, there are too many, you know, particles or the concentration is too high, I tend to collect 1 image in the center. The throughput overall is, yeah, a little bit faster with more images. Yeah.
Thank you.
Sure.
All right, there are a few more questions in the chat. Patrick had one. Did you want to ask that yourself, Patrick, or?
Well, it's not a very important question. The hexagold grids, do you think they'll make much of a difference?
I hope they will. You know, they're still the gold grids. I had a chat last week at the Gordon conference with Katerina and Chris Russo. You know, they were both there, and Katerina gave actually a talk about the gold grids. So what she said is, with these grids, they seem to get very uniform eye thickness across the hole. So if that's true, of course, it depends, of course, the sample, the concentration, and so on. But if we can get even more consistent and reproducible sample prep, of course, that will help. In terms of motion, you know, the gold grids we're using now already pretty good. Again, I don't want to say that will not have an effect, but maybe we can go to 1.5 angstrom GPCRs in the near future. I don't know.
Let's get there. Thanks.
Or sub 2 angstrom receptor only.
All right, I think Melissa also had a question if she wants to ask that herself, or else I can read it.
Thanks, Rado. That was a fantastic talk. Particularly, I enjoy your technical detail, which is sometimes missing in some of these talks. I just had a question about your GPCR complex in complex with the nanobody 35. You said that it gave improved resolution because of the improved stability. I was wondering how you pre-screening those nanobodies for stability enhancement. Are you using HDX? Are you doing something else?
You should probably ask Patrick about this if they pre-screen, but in a sense, I think there was also motion analysis. I don't remember all the details, sorry. But yeah, just from the cryo-EM data and the dynamics, after processing of the data, I think the nanobody definitely stabilized and make it more rigid. So
Okay.
About the biochemistry, maybe Patrick could chime in.
Okay, sure. Thanks.
Cool. Gokhan Tolun had a question.
Hi, Rado. So what I was wondering is, the new energy filter selectors is released now. So what is your take on using a selectors versus a more conventional or the energy filters that are available already in many places?
For us, in biology, as long as the filter is not terribly misaligned, the distortions are non-performance limiting of these filters. So both filters are energy filters, basically. The way things company like to presenting is we invented the energy filter. No, energy filters existed long before and will continue to exist. So basically, they're both filters. It comes down to the ecosystem you subscribe to, Gatan or the other one, and the overall system performance. So one thing about the integration is that your system is really one, and it's all designed to work together, works with the same software and so on. So Selectris and Falcon 4 is a wonderful combination. I recommend it to all the new, the places that want to have their first microscope or that are not as experienced. Of course, in terms of stability, I believe what they claim that they have very stable, zero loss alignment. And I don't know how it holds up if it's in an unfavorable EMI environment, like in our case with the microscope near the elevators. But other than this, yeah, it's a wonderful combo. And if you have any issues with it, the thing is you have one number to call. They come and they fix the whole system, and it's all made to work together. If you have two manufacturers, supplying different parts, there are always issues related to service, to compatibility. Like in our case, this issue with the first frame having the pattern noise is partially related to the fact that the K3 has to be integrated with the EPU. And because of this integration, they have to use a different version or something, and that messed things up. So, I would definitely want to have a cohesive and, single system. On the other hand, we want to have competition, right? We want to have choices. Basically, as a user and as a customer, we should be able to select between several options, hopefully, and also for the microscopes, not only for the cameras. And that's the real democracy in cryo-EM, not having a few choices from one manufacturer. The real democracy is when you have several candidates from different parties, not several candidates from one party, right?
Yeah, great. Thank you.
Thanks, Gokhan. It's good to keep going. Rado? Rado, are you good to keep going with questions for a little bit?
Of course, I'm at home. I basically work from home, but I have nothing urgent, no experiments today.
Thank you. Alistair?
Hi there, great talk. Sorry if you already answered this, my internet briefly cut out. But with the gold grids, you always get these bright spots over the place, and you briefly mentioned you were going to try and reduce those by using a different aperture maybe, but how detrimental, how bad do you think these bright spots are? Do you think that the particles included in the final reconstruction, are they causing more subtle changes than that?
Yeah, surprisingly, they are not that detrimental. So when I compare the objective lens aperture versus no aperture, so the aperture dataset, which used the 100 micron objective lens aperture, didn't have any spots. Basically, as long as you're not very close to the edge, collecting very close to the edge, you don't see the spots if you put the aperture in. Of course, be very careful to center the aperture well, because otherwise it can introduce terrible astigmatism or higher order aberration. Apart from that, the processing of the datasets, even some of the datasets who reach sub 2 angstrom, they were collected without an aperture, and you would see there these spots. But what they would do is the areas where the spot is, you have more electrons basically that are not contrast carrying or information carrying, so they would just add more noise to the background. So these particles are more noisy, I guess, and they probably get classified out during the classification steps. But there are not that many spots, there are, here and there, maybe one spot per image.
So a few percent, but you're just kind of losing a few percent of particles rather than putting
probably, yes, a few percent. Yeah, that's a good guess. Yeah.
Thank you.
Sure.
Jason, did you want to ask your question or do you want me to read it for you?
Yeah, I can ask a question. Thank you, Rado. It's really a fantastic talk. And I think you mentioned in your previously CGRP receptor dataset, I mean the one without G protein, the well-packed particles will give the helpful hand to the eye thickness. However, in the following, like the similar receptor-only dataset we gained from your lab in Japan, or we did the image in Melbourne, we got some problems on the preferred orientation of particles. I think because without G protein, this receptor only in my cell prefer to show the top view instead of a mixture of top view and side view. And that gives quite a big issue on the following processing. Do you have any comments on suggestion for solving this issue?
Yeah, preferential orientation is the killer nowadays, I would say, for a well-purified particle. So definitely, even with the CGRP, there was preferential orientation. There was, if you look at the maps, they definitely have an isotropic resolution effect on them. There are a few solutions. One is to collect data with tilted grid. I haven't tried that. We haven't tried it. I guess many people resort to that as a measure to resolve issues with preferential orientation. With the receptor only, it's already on borderline in terms of will it reconstruct or not, as you probably know, processing these data sets. So tilting the grid may actually kill it. I don't know. We have to try. The other possibility is to try different detergents, like screen a few detergents and see if anything makes the particles stick less to their water interface. I don't know why the receptor-only structures tend to have this higher. In general, smaller particles tend to have more preferential orientation issues, probably related to their overall, what is that, fluid behavior in the fluid, basically fluid dynamics and how often they bounce off the surface. So basically how many times they hit the surface before the grid is frozen. Just try either tilting the grid or several detergents, maybe in addition to the detergent that was used to solubilize the particles, maybe add a little bit of a different detergent or a polymer like PEG or something. I don't know.
Okay. Yeah, we'll have a try. Thank you very much. Yeah.
All right. Does anyone else have a question they'd like to ask? You can just pop your hand up, or else we might leave it here for today. Doesn't look like we have any more. Thank you, Rado. And thanks, Patrick.
No, I'm just popping back to say many thanks to Rado for a brilliant talk. Enjoyed it a lot. Lots of interesting questions, so that was fantastic. And yeah, definitely when we can move around between Japan and Australia without quarantining, we'll fly you down for sure.
That would be great.
Okay.
Thanks for coming and thanks for hanging around to answer questions.
Cheers.
Well, it was a lot of fun. I like questions, especially when they're related to the experiment. Okay.
Cheers.
So thanks for having me, and see you sometime in person.
Brilliant. Thank you. See you later.
Bye.
Thank you.